Embryonic Stem Cell Research by Arnav H., Trisha P., Safa B., and Zad N. The following is the summary of the case. A couple, Mr. and Mrs. Smith, were trying to have a baby, but they couldn't have it naturally, so they went to an IVF clinic. During their visit to the clinic, the doctor was successfully able to fertilize 8 of Mrs. Smith's eggs. 3 were implanted into Mrs. Smith, 2 were given birth to, and 1 survived. The remaining embryos were kept safe for possible implantation for later on. The Smiths died due to a car accident, and the frozen embryos were left at the hands of the clinic. The clinic decided to donate the embryos to a research team that was researching Parkinson's, which is a progressive brain disease. The issue here is, was the clinic doing the right thing by donating someone else's embryos without their consent for research? The ethical concern about this case is that alternating and performing experiments on a frozen embryo is wrong because the embryos have a possible chance of becoming a living organism. The social concern about this case is that some people might not want others to donate their embryos for research because they think it is ethically wrong to give away their potential child. The legal concern about this case is that scientists might not be able to obtain the resources and materials needed to do the research from the government because government officials might think that this research is ethically wrong.
So our position on the case. Our group believes that stem cell research is worth the potential benefits, meaning that the embryos should be delivered to the scientists so they could study a cure for Parkinson's disease. Additionally, our group believes that the clinic in the case made the correct decision by using the Smith's embryos for science. This is because of the following reasons. The clinic did not know what the Smiths wished to be done with the stem cells if they were not implanted. This means that the Smiths didn't give any inherent will for their embryos that specified a specific person, meaning that they could be used in any other research. Also, the country the clinic in had no legislation or laws regarding stem cell research, meaning it was legal to use that research in the country. We also believe that stem cell research should be further differentiated into researched as they can differentiate into many different and specialized types of cells, such as cardiac cells and bone cells. This means that stem cells can be used to cure certain disease since they can differentiate, causing them to replace dead and damaged cells. Lastly, they can also test the safety and effectiveness of new drugs, which could also profit the community by helping prevent further diseases. Now I'll delve deeper into stem cells. What is a stem cell? A stem cell is a cell that can become a more specialized kind of a cell, meaning they differentiate into different types, such as blood or cardiac. The 3 types of stem cells regarding the amount of specialization are totipotent, pluripotent, and multipotent stem cells.
What are embryonic stem cells? Embryonic stem cells are totipotent and pluripotent stem cells. These cells are totipotent before the formation of the germ layers, and these cells can differentiate into any type of cell. They are pluripotent after the formation of the germ layers, however, and are slightly more limited than totipotent stem cells in what they can become. These germ layers are the ectoderm, the mesoderm, and the endoderm. In this image, one can view the 3 germ layers. The ectoderm develops into the outer part of the organism, such as the skin and hair, while the endoderm develops into the digestive system. These 2 germ layers developed before the 3rd one, which is the mesoderm, and in between the 2. The mesoderm makes up the other parts of the body, such as the skeletal muscle, bone, and connective tissue once these stem cells have developed. What are embryonic stem cells? Continued. These cells derive from the blastocyst, which is the fertilized egg for mammals, and a rapidly growing cluster of cells. This is called a blastocyst 5 or 6 days after fertilization. The inner group of cells of a blastocyst will become the embryo, while the outer group, the blastocyst itself, serves to protect and nourish this embryo. During this time, the embryonic stem cells only consist of about 50 to 100 cells. Embryonic stem cells can become any type of specialized cell, including nerve, liver, cardiac, brain, bone, and blood cells. In this image, it is shown how stem cells can and do become cardiac, blood, liver, and nerve cells, although there are more that they can become, as said above.
What is an adult stem cell? Adult stem cells are stem cells that can differentiate into different types of cells, meaning that they're blank slates that can specialize into specific cells. Unlike embryonic stem cells, they can only differentiate into certain cells. They are found in your body after it has fully developed. They replenish dying cells and replace damaged tissue. They are commonly known as somatic cells. These are any biological cells that form in the body of an organism, almost any cell other than a gamete. A gamete is a sex cell used in reproduction. These cells can be found in any human and therefore can be extracted from any donor. These are unlike embryonic stem cells, which can only be extracted from female donors. Adult stem cells are limited in what they can become. They are less versatile and more durable, but they are also more likely to contain abnormalities.
Induced pluripotent cells are also known as iPSC. These cells are derived from skin and blood cells, and they are somatic cells that are reprogrammed into their pluripotent state, which is very similar to the embryonic stem cell. The reason why scientists don't use them is because they can cause harmful health conditions such as cancer, and these cells are able to cause cancer because they use retroviruses to program the cell. Retroviruses have a chance of inserting unnecessary genes that can cause tumors. Even though the possibility of these cells causing adverse health effects are unknown, it is proven that the chances of the cell being rejected by the immune system are very low. Perinatal stem cells are also known as amniotic stem cells. There are more studies that need to be done since scientists don't know a lot about these cells.
How does stem cell technology work? The process begins when the embryos are extracted from a female. They are then sent to a lab where they will be fertilized there rather than the female they were extracted from. This is known as in vitro fertilization, or IVF. These fertilized stem cells are pluripotent and can give rise to any type of cell in the body. This is done through the use of hormones. These cells have not yet differentiated, which is partly why they can become any type of cell. They can also create copies of themselves indefinitely. The technology uses this aspect too when they are exposed to said hormones. They divide, and with each division, the daughter cell becomes more and more specialized for that specific cell. When the embryonic stem cell has fully specialized, the cells will stop dividing. Over here, in the first image, you can see how scientists collect sperm and fertilize eggs manually. In the second picture, you can see how the in vitro fertilization process occurs and how it leads to the cultured pluripotent stem cells.